It's officially 12 o'clock, so I can say good afternoon, everybody. Welcome to our CERI seminar series, our hybrid event on site at BMRI and the Tygerberg campus of Stellenbosch University, and online to over 30 at the moment and climbing participants. Whoop! See how special you are. Over 50. Yeah. Those over 50, if you happen to be in BMRI, come down and join us. And there's food outside as well. Let me just add that in there. So welcome. I'm Lenine Liebenberg. I'm based at CERI, and it is my absolute pleasure to be inviting our special guest today, Dr. Seth Bloom, MD, PhD. As you can see, I'm not Tulio de Oliveira, who usually does the introductions for these meetings. He said, no, it's your guest, so it's your turn to embarrass him before he gives the talk. So I'll try my best. But I think he's got one step ahead of me. He put a picture of me on the front of his, his presentation. When Seth said that he'll be coming through to join us, I asked him, so what do you want to do in Cape Town while you're here for this one day? He said, Table Mountain. Can we get up to Table Mountain in the quickest, most strenuous walk possible? And I said, with me? Are you sure? He said, yes. So there he is dragging my bulk up to the top of Table Mountain yesterday and standing here wobbling in front of you guys. But we made it. And he doesn't look any worse for wear, ready to have this very special presentation with us here today. So the point of these series seminar sessions are to have really great, world-renowned, high-class scientists coming through to visit us, share their ideas with us share our ideas with them, see how best we can work together. Another part to address the world's most pressing problems. Another part of these sessions is to encourage early career researcher development. So I'm sorry about those participants online. If you happen to be within the vicinity, please do come down on site to the BMRI auditorium. Seth is available after the session, after a little mingle, back in this room, for a little very casual Q&A. You can rip him to pieces. You can ask him anything you want. What's your favorite color? How did you get to where you are? What tips could I use to get to, MD, PhD level? What is your, okay, I'm not giving you any more ideas here. Okay.
How can I get a position in Harvard?
Woo! Right, so Seth Bloom.
And should you?
Sure, good question. Seth is a physician scientist. He's based at Massachusetts General Hospital. He's, what's that? Also within Harvard. His background is in, if you're interested on a cervicovaginal microbiome and its metabolites and how to manipulate the genital microenvironment for better health outcomes for women. And he'll tell us all about that as soon as he quickly passes that picture of me to the next slide. Welcome very much, Seth Bloom. I'm so pleased you're here. I'm going to wobble down over there now. Okay.
Well, can everyone hear me well? Okay. So thank you very much, Lenine, for the invitation, and to CERI for hosting me and for tolerating my decision to go up Table Mountain. I'm not sure if she'll ever invite me back again, but at least we've done it once. So today I'm going to be talking about identification and characterization of novel therapies to modify the vaginal microbiome. I have no financial disclosures or conflicts of interest. And so I'm going to just give an outline of the talk. I'm going to start by giving some introduction into some basic paradigms in bacterial vaginosis and vaginal microbiome research. I'm then going to talk about what we know or think we know about vaginal microbiota dynamics in the context of current therapies for BV. I'll talk about some unpublished research on the microbiome effects of a live biotherapeutic product, or LBP, for bacterial vaginosis. I'll then talk about a little bit about some published work we've done on methods for studying and selectively targeting a species called Lactobacillus iners and explain why we think that's important. And then I'll talk about an unpublished, although some of it is pre-printed, study on Lactobacillus oleic acid metabolism as a novel target for BV therapy. I want to start off by introducing everyone to some basic paradigms in bacterial vaginosis and microbiome research. I know we have certain people in the world who are world famous researchers in this area, and others of you who I assume are not particularly familiar with this area and work in other fields. And so I want to make sure that for the course of the discussion, we're kind of understanding the background, the terminology, and the sample types that we're dealing with. So with apologies to those who already know this by heart, I first want to just start off by talking about some common sample and data types that you'll hear in this presentation and other literature in the field. And so basically, when we're talking about research on the vaginal microbiota and related immune phenomena and so on, usually what we're talking about is studies of mucosally collected samples from the cervix and vagina. And these are typically collected either by a clinician or sometimes self-collected by a study participant on a cervicovaginal swab or possibly a cervicovaginal lavage or a cervical cup that can collect vaginal or cervical secretions. Once the sample is collected, there's a variety of things that can be done, and you'll be hearing examples of all of this throughout the presentation. So one obvious thing is we can do sequencing. So we can isolate DNA or RNA, and we can profile microbiota composition via 16S sequencing, via shotgun metagenomics. We can do taxonomic assignments. We can look for presence of specific genes within the metagenomes. We can also generate what are known as metagenomically assembled genomes, meaning rather than isolate the bacteria and sequence their genomes, you attempt to use the shotgun metagenomic data to assemble a bacterial genome, and examples of that will come up throughout the talk. You can also use the material collected to do assays for various soluble molecules like metabolites, cytokines, and so on. So these samples can obviously be used for clinical testing for sexually transmitted infections, just like you would get if you went to your doctor to be screened for chlamydia or something, or for gram stains to assess for bacterial vaginosis diagnosis. And then one thing that'll come up a lot in the second half of the talk is we can do primary bacterial isolations from these samples. We can isolate various types of species in clinical samples. We can study them, we can sequence their genomes, and we can do experiments with them. So again, all of these modalities will be putting in their appearance at various points during the talk. So what is bacterial vaginosis? I keep referring to it. BV, for those who don't know, is the most common vaginal discharge syndrome worldwide, at least amongst premenopausal women. And it's really seen throughout the world, and it has high rates in pretty much any population you can look at, but disproportionately in a wide variety of societies, cultures, geographies, it has been shown to disproportionately affect women with low income or education levels within societies, tends to impact members of racial or ethnic minority groups across many different populations. And although it is widely present in many parts of the world, there are a number of studies that suggest it's really disproportionately prevalent amongst many African populations, particularly in Eastern and Southern Africa. We'll talk very extensively about this next point throughout the rest of the talk, but it is associated with a low lactobacillus vaginal microbiota. And not everyone who is diagnosed with BV is necessarily symptomatic, but many people are. And the characteristic or canonical symptoms associated with BV include vaginal discharge, a so-called fish-like odor from volatile biogenic amines produced by the microbiota. People can experience symptoms like vaginal itching, pain, or burning, and sometimes burning with urination. And one key thing to know about bacterial vaginosis is that it is associated with inflammation and with alterations in mucosal immune responses. So this is a really important foundational paper that was done just down the road by Joanne Passmore, Lindy Masson, and colleagues at UCT, looking at cervicovaginal cytokines in women here locally who had a diagnosis of BV. And what they observed, and this has been replicated in many other studies, including some of our own, was that there were really key differences in certain inflammatory cytokines. For example, IL-1 alpha and IL-1 beta were much elevated in women with bacterial vaginosis. So that's the clinical syndrome of bacterial vaginosis. How does this relate to microbiota or microbiome composition as we might think of it in kind of the modern sequencing Freudian slip, era? Okay, so to introduce you to some of the key concepts that will again be coming up through the talk, I want to go back to really probably the foundational modern next generation sequencing study of the vaginal microbiota. This is a paper from 2011 by Jacques Ravel, and this was a population living in the US in Baltimore. And they did bacterial 16S ribosomal RNA gene sequencing and made a number of observations, many of which have stood the test of time. And so I'll just kind of walk you through this here. So let's see, is my mouse showing up? Okay, can people see my mouse? Yeah. Okay, great. So what we're looking at here is a figure where each column is a different individual, or a different sample, I should say. And each row in this middle block is a different bacterial taxon. And then down below, we have the alpha diversity of the samples, which those of you who do a lot of microbiome work will know what that means. Up above, we have people's Nugent scores, which is a way of measuring whether people have bacterial vaginosis, and also their vaginal pH, which is closely related to bacterial vaginosis as well. And so what was observed here? So in this heat map, if the color is red, that means there was a lot of this taxon, and if the color is yellow, it means it either wasn't seen or was barely seen. And so what you can see if you look through here is these kind of blocks or clusters of people. And if we start right here, we have this block of people who had a very high relative abundance of this species called Lactobacillus crispatus. And when I say high abundance, I mean this could be up to greater than 99% of all of the bacterial reads seen within the microbiota. So very Lactobacillus predominant microbiota. And they referred to these as community state type or group number 1. There was another Lactobacillus species, Lactobacillus iners here, which had a similar phenomenon, could be very highly abundant, was relatively non-overlapping with Lactobacillus crispatus. In their nomenclature, they referred to this as community state type 3. There were a couple of other lactobacilli that more rarely could occasionally dominate the microbiota, and this included Lactobacillus gasseri here and Lactobacillus jensenii, and we now know this would also include a species defined as Lactobacillus miliaris. These are names you're going to hear throughout the rest of the talk over here. And then finally, there was this group that they referred to in their nomenclature as community state type 4, which was really not dominated by any lactobacilli. So these are lactobacillus deficient microbiota. They're much more diverse, so there's a lot different taxa here. And these are largely anaerobic species of various sorts. And these are the people, if you look up here, who have high Nugent scores. So these are the people who have diagnosis of bacterial vaginosis. They also have high vaginal pH because they don't have lactobacilli making lactic acid. And so, as I said, this was one study from 2011, but it's really borne out quite well in many, many different studies in many, many different populations. And so, although people have used slightly different taxonomy and nomenclature and so on, so for example, here is a paper that was published from my postdoctoral mentor's lab several years ago, Doug Kwan's lab at the Ragon Institute. And this was working with a cohort based in KwaZulu-Natal, just outside of Durban, of a group of young, otherwise healthy women in a high-risk HIV community. And there's a very similar pattern that you can see here. So again, each column is an individual, and this is stacked abundance bar plots, but each color is a different taxon. We have a group of folks here who have high Lactobacillus crispatus levels. We have, in the Kwan lab nomenclature, which is slightly different, this is cervicotype 1. We have a group of people here who are, again, Lactobacillus iners dominant, referred to as cervicotype 2 in Kwan lab publications. And then here, the non-lactobacillus dominant microbiota has been divided into 2 groups, one of which is referred to as cervicotype 3, which has a lot of members of the Gardnerella genus. And over here, we have basically, quote, other, but these are in general diverse anaerobic communities. And once again, these people over here are very closely associated with having BV, and being over here, few or none of these people have BV. Okay, so now that I think we're hopefully got some of these principles planted in mind, What about the associations of the microbiota and bacterial vaginosis with actual disease? So first off, I think we need to be very explicit that BV, although it doesn't get a lot of press, is at least a clinical syndrome, if not a disease. And it has really important impacts for many of the people who experience it. It can affect, if you're symptomatic, quality of life, intimacy, self-esteem, sex life, and psychosocial well-being. And this is some quotes from a study of women and transgender men in Australia who'd had BV or recurrent BV. And one of them just says it was just a very horrid discharge and it smelt and it just felt unusual. And another one who'd been experiencing recurrent BV says it's not debilitating physically, but it's debilitating mentally and emotionally, because even though this affects many, many people in many, many different societies, the symptoms of this condition are such that it's very difficult to talk with people about in many cases. But aside from those impacts and those psychosocial impacts, there are also really important associations with other diseases that get a lot more press. And so, for example, HIV. So this is a meta-analysis of a study that looked at people with or without bacterial vaginosis and the risk of subsequently going on to contract HIV. And what was observed here in these studies and also in the meta-analysis of these studies was that having BV was a substantial risk factor for going on to contract HIV. That's based on the clinical diagnosis of BV. What about specifics within the microbiota composition? So going back to this study published from the Quan lab several years ago, we talked about these different cervicotypes, and what was observed in this study, the study had enrolled HIV uninfected women in this very high-risk community. Unfortunately, many people over time did go on to contract HIV, none of whom were in the group colonized by Lactobacillus crispatus, at least within the course of this particular study in this cohort, whereas having Lactobacillus iners dominance or these non-Lactobacillus dominant states was associated with substantial rates of HIV acquisition. And again, these are prospective risk studies, but it's not just that. There are associations with many other outcomes of clinical importance. So preterm birth has been associated with microbiota composition and BV. Many aspects about something very near and dear to Lenine's heart. So human papillomavirus infection, persistence, development into cervical cancer, and even some recent studies suggesting that specific microbes within the microbiota, possibly specifically Lactobacillus iners, may have an association with poorer responses to cervical cancer therapy. Also associations with sexually transmitted infections for a variety of hypothesized reasons, peripartum infections, and mucosal inflammation. And I should mention, I'm not really gonna talk a lot about the immunology or the immunity or many of these other conditions during the rest of the talk. I'm really gonna focus on the microbiota itself. But this is a rationale for why we wanna understand this. We think many of these relationships are causal, not just associative. There's a variety of reasons to think so. And so by focusing on the microbiota and what can be modified about it, we hope that this could have important impacts on the immunity and many of the disease states that we're talking about here. Another thing to be aware of within this field, however, is that there have historically been some very important barriers to progress within the field. So this is a field that really lacks robust animal models, which is a huge challenge for studying diseases. And the particular issue is that humans really appear to be the only species that can be colonized stably by these vaginal lactobacillus species. And so that is a problem that has largely not been solved in a robust way to facilitate experiments. Secondly, many of these vaginal bacterial species have either never been grown in culture or are very challenging to isolate and cultivate and study in culture. And that's obviously a huge problem if you're trying to discover mechanisms and therapies, although we'll come back to this for at least one of them later in the talk. And this is something that I've done a lot of work on. There are also historically no tools to genetically manipulate these or most of these species, which is again very important if we really want to understand mechanisms that are going on within the vaginal microbiota, although again we'll come back to that later in the talk. So to move from kind of this basic background to get more focused into what we're really going to be talking about today, what happens within the vaginal microbiota over time, particularly in the context of people who are being treated for bacterial vaginosis, which is the current example of what we do to try to therapeutically modify the vaginal microbiota. So how do we treat vaginal BV, and do these treatments work? So first-line treatments, WHO approved, CDC approved for bacterial vaginosis, all involve antibiotics. Largely, these involve the antibiotic metronidazole, which, as many of you will know, is broadly active against anaerobic bacteria and has no intrinsic activity against lactobacilli. And the idea here is you're killing off these BV-associated anaerobes, and hopefully your lactobacilli will take over. So does this work? Well, the answer is yes-ish. So many people do receive at least transient benefit in a wide variety of studies, but this is a large study of women with symptomatic BV in Australia, although it mimics findings from many other studies around the world. And many of these women received an initial benefit when treated with metronidazole, but then we can look at, this as survival, but just they weren't dying, don't worry. This is just time to recurrence of recurrent bacterial vaginosis. And what you can see is that within 1 year of therapy, over half of these individuals had gone on to experience recurrence of their bacterial vaginosis. So this does work, and targeting the microbes does work, but it's not working nearly well enough for nearly enough people. And many people suffer chronic or recurrent BV. And here's again a quote from this prior study I mentioned, where we have a young woman who says, I already accepted the fact that I have BV. I already kind of accepted that I'm not going to get rid of it. So this is someone for whom current therapies just aren't working. And as a consequence, she's at risk for many things in the future, potentially. So what's going on within the microbiota when you give someone metronidazole therapy? So this is kind of an early study looking at this from Sujatha Srinivasan and David Fredericks in Seattle. And this is a profile of a single patient who had recurrent bacterial vaginosis and was treated, and they used a qPCR-based technique to look at the abundance of certain key bacterial species. So early on in this person's first episode of BV, they had various BV-associated anaerobes. They were treated with metronidazole. Those went away, and here in the back, we have an expansion of Lactobacillus iners. So that continued for a while, and then they recurred. The iners went away, and all these BV-associated bacteria came back, and they had clinical bacterial vaginosis again. They were treated again. The iners came back. They were free of bacterial vaginosis. Then they had another recurrence. The iners went away. These BV-associated anaerobes came up, and then they were treated again, and the iners came back again. So this is, anecdata, as we say. This is one person, but it turns out that this is a pretty consistent pattern if you look at what happens with people who are treated with metronidazole for BV. Sometimes you don't get rid of these bugs at all, but very rarely do you get Lactobacillus crispatus dominance. So for example, here are two studies, one of which was conducted in Kenya on the upper right, and one of which was conducted in the US on the lower right. And in the study in Kenya, for example, when women were treated with metronidazole for asymptomatic BV with the idea that it might mitigate HIV risk, the immediate effects of treatment, or the within a month of effects of treatment, were largely an expansion of Lactobacillus iners rather than Lactobacillus crispatus for most individuals in the population. And in this US cohort of women who were treated for BV, a month later, essentially all of them were colonized at varying levels with Lactobacillus iners, but only a minority had colonization with Lactobacillus jensenii or crispatus. So it seems like crispatus is indeed, associated with the most optimal health outcomes in a variety of different studies for at least many different health outcomes. And yet this is really not what we're achieving when people are being treated with standard first-line care. And so getting into some more details of kind of some unpublished research that we have, one of the studies we've been working on is looking at the microbiome effects of a live biotherapeutic product containing Lactobacillus crispatus for treatment of bacterial vaginosis, or a prevention of recurrent bacterial vaginosis. And I just want to highlight, I'm going to have an acknowledgement slide at the end with all of the people, including all of the professor level folks who contributed to this research, but I really want to highlight as I go through for our research some of the people who are kind of in the lab, in the trenches, doing a lot of the work, whose names don't necessarily appear at the top of the website. So first off, this is a study where the co-lead author along with me is Laura Simmel, who started this as a postdoc at Stanford and is now continuing to work on it as an assistant professor at UC Leuven in Belgium. I'm actually not going to talk about Laura's analysis, which is ongoing, but we've been working very closely on it because I'm only going to talk about a little part of this work. And then within the lab, I just want to highlight some people who've really contributed. So Joseph L. Sherbini is a bioinformatician in the lab from the US. Jia Wu, originally from China, is the lab sequencing manager and has done all the sequencing I'm going to talk about. Selena is a young woman who grew up in Zambia, who is a technician in the lab and now applying to medical school. Ashley is a former technician in the lab from Zimbabwe who's now gone into biomedical consulting. Jonathan is a dry lab technician in the lab from the US who's applying to PhD programs. Aditi's another US-based technician who's going into a PhD program, and Fatima's a postdoc who's about to start an independent faculty position at Tufts University. So the project that we've been working on looks at samples from this really landmark study of a live biotherapeutic product called Lactin-V to prevent recurrence of bacterial vaginosis. And just to be very clear, I was not directly involved in any way in the actual conduct of this trial, though Craig Cohen and Anke Hemmerlin, the first and last authors on this paper, are collaborating with us on the subsequent analysis that we're doing. But the original paper was published a few years ago in the New England Journal of Medicine. And so it employed Lactin-V, which is a single strain live biotherapeutic product containing the Lactobacillus crispatus strain named CTV05. And this strain is prepared as a freeze-dried powder along with a preservative and is self-administered or clinician-administered via a vaginal applicator, of which we're seeing a picture here, at a dose of 2 × 10⁹ colony-forming units per dose. And the placebo for this trial was the same preservative administered the same way, but without any bacteria in it. So what was the design of this trial? In the design of this trial, participants all had bacterial vaginosis diagnosed at baseline. They were then treated with intravaginal metronidazole gel, which is a standard first-line treatment. And then they returned within 48 hours of stopping their antibiotics. They returned to the research clinic. And at that point, they were enrolled and randomized in a 2-to-1 ratio to either receive Lactin-V or the placebo for prevention of recurrence of bacterial vaginosis. And so they had a sample collected when they had BV, immediately post-treatment before they'd started this product. They then came back multiple times for different study visits at which samples were collected. And if you were enrolled in this trial, you self-administered this to yourself for 11 weeks, multiple times per week. And then at week 12, so shortly after stopping therapy, that was the primary endpoint for the study. And what they were looking at was whether you'd had recurrence of bacterial vaginosis during that time. And then the secondary endpoint was at week 24, which was so 13 weeks after you had stopped the therapy, and looking to see whether they had recurrent bacterial vaginosis. And so what were the outcomes of this? Well, it worked. This was a phase 2b trial, and there was a signal for efficacy, meaning there were lower rates of recurrent bacterial vaginosis in the folks who got Lactin-V rather than the folks who just got standard of care plus placebo. So this was very exciting. And since we don't have an animal model, in addition to being a therapeutic advance, it's also the proof of principle of many of the things we have thought we have known about Lactobacillus crispatus and its role in prevention of recurrent BV. However, if you look at these numbers really carefully, and this is just a graph I made replotting them, you can see that this was a success, but it wasn't quite a smashing success in the way we might like. So what we're looking at here is just simply the numbers from this table replotted. And here we're looking at week 12 and week 24. We have the Lactin-V group on the left and the placebo group on the right. What you want is not to have recurrent BV. What you don't want is to have recurrent BV, and that's shown in green. And what you can see is there's a lot of people who had recurrent BV, even in the treatment arm, especially by week 24. There's just a large fraction of people who still didn't really get the benefit one would have hoped from this therapy, even though it was better than standard of care. And so where we came into this was to try to say what was actually happening within the microbiota that might give us insights into why this occurred and how this treatment was effective. So we got swab samples from all these different studies, study visits, pre-metronidazole, post-metronidazole, weeks 4 and 8 when you're on treatment, week 12 when you just stopped, and week 24 when you've been off it for 3 months. And we generated many types of data, some of which I'm not going to talk about today. So 16S ribosomal gene sequencing, shotgun metagenomics. We've done some metagenome assembled genomes and strain tracing, which I won't get into in detail. We generated a closed genome for the live biotherapeutic strain to better guide some of this work. We have isolated, again, I probably won't have time to get into it, but genome sequenced isolated bacterial isolates from study samples, done cytokine profiling, and we have a variety of clinical data and so on. But I think the really top line finding that I want to bring to you here is looking at specifically what happened to the microbiota within people in the Lactin-V trial. And for this analysis, as a study team, we decided to really focus on the question of people were being administered Lactobacillus crispatus, so we could look at people from the perspective of, did you achieve high-level Lactobacillus crispatus colonization, greater than 50% of your microbiota, as one category. Alternatively, you could have less than 50% of total Lactobacillus of any ilk, and that was the condition down here in the blue. And then that, by default, leaves us this condition of people who have a lot of lactobacillus, but it's not predominantly Lactobacillus crispatus, which usually means that it's Lactobacillus iners. Okay, so defining these categories as kind of endpoints that we can think about for what was happening from a microbiome perspective, we can then look at what was happening in the treatment arms here. And so if we look here, what we can see is that at enrollment before metronidazole, when everybody had bacterial vaginosis, not surprisingly, almost everybody here is in the blue, meaning they had a non-lactobacillus dominant microbiota, which is what you would expect. Immediately after they stop therapy, before they've gone on any other sort of study product, the majority of people now did have lactobacillus dominance, but it was not Lactobacillus crispatus in almost all cases. Usually this was Lactobacillus iners. There were some people who still had a dominance by BV-associated bacteria as well. And then by week 4, now people are either taking Lactin-V or they're taking placebo. And at this point, you can see the study arms diverge, right? So here's placebo and here's Lactin-V. And at this point, we have a decent fraction of orange up in the Lactin-V arm, and that perpetuates throughout, and very, very little orange, meaning very, very little high-level Lactobacillus crispatus colonization down below in the placebo arm, which is consistent, perhaps even more definitive than a lot of the prior studies that I was showing you in terms of what was going on. And if we look at week 12, which was kind of the New England Journal time point that they used as their primary endpoint, and we say what's going on here, you can see, so we have the Lactin-V arm on the top, we have the placebo arm on the bottom, and there's a lot more people in the Lactin-V arm who have high-level Lactobacillus crispatus colonization. There's people in both arms who have Lactobacillus iners dominance, and in both cases, almost none of these people had a clinical diagnosis of BV at this point. And then over here, we have non-Lactobacillus dominant microbiota. Many of these people also met clinical criteria for BV, and these were various mixed BV-associated, mostly anaerobes. And this is not surprisingly, as you can kind of see by eye, these differences are statistically significant in terms of Lactobacillus crispatus colonization. So there's over a 3-fold benefit at week 12 and even higher at week 24. For having a crispatus dominant microbiota in the treatment arm versus the placebo. However, again, the other thing you can see, so these are people who have been giving themselves 2 × 10⁹ colony-forming units of live Lactobacillus crispatus multiple times per week for weeks and weeks, and the majority of them were not colonized at high levels with Lactobacillus crispatus. So this both explains why this treatment works better than placebo, and it also gives us insights into why the effects are only partial. For many people, the Lactobacillus crispatus is just not establishing itself, and that really seems to explain the incomplete efficacy. And so what can we say stepping back from all of this? We can say that, or at least one could argue, that we think what we want are added interventions or synergistic interventions that could selectively inhibit L iners and or selectively promote L crispatus and or help us better target BV-associated bacteria, including some that may be metronidazole resistant. So can we get ourselves to these endpoints? And this is where I'm going to kind of shift gears from these human-based, population-based studies, clinical studies, and really get into the bacteriology that we've been doing. And so the first part of what I'm going to talk about is based on a published study, which I'll just actually talk very briefly, not going through most of the findings, where we started really trying to look at Lactobacillus iners a number of years ago. And I just want to highlight that folks who were involved in this, this part of the work. So Namfu is a former technician in the lab from Botswana. She's, some of you who are involved in the Vibrant study now know her because she went on to do a master's of public health at Harvard and is now a project coordinator for that study. Matt Hayward is a former technician from the UK who's now in industry. Aaron is an undergraduate, or was an undergraduate working on this project with me. He's about to start an MD PhD at Stanford. He's from the US. Jo's a young woman from Ghana who worked with us as a technician for a while, is now an MD PhD student. Jawu, and you already met. Nandu is, I think I mentioned, but is a career technician based in Durban. And I already introduced Fatima. So what's up with Lactobacillus iners? Okay, so when I started my postdoc, people were writing reviews with titles like this: Lactobacillus iners, friend or foe, or Lactobacillus iners, the unusual suspect. And I want to highlight that these were reviews. These were not research articles. And in fact, back in 2018, here in Cape Town, there was a Keystone Vaginal Microbiome Conference that was co-organized by Joanne, that was attended by top researchers in the vaginal microbiome field from around the world. And of all the studies that were presented there, there wasn't a single experimental study using Lactobacillus iners, which is crazy because Lactobacillus iners is the most consistently abundant human cervicovaginal species worldwide if you look across many studies. Almost everyone has at least a little bit of it, and there's a fraction of patients in any, or women I should say, in any population who would tend to have high levels of Lactobacillus iners. It's associated with a higher vaginal pH than other lactobacilli. It makes lactic acid in different ways than many other lactobacilli. It has a putative virulence factor, so it may actually be directly pathogenic, or it may be less protective. This is debated. It was known at the time that it had a reduced genome size and it had lost many metabolic genes that was suggestive of niche adaptation for the human urogenital tract. But it was really not being studied well because it fails to grow. This was a species-defining characteristic, that it fails to grow in standard Lactobacillus MRS media. And because of this, despite the fact that this is a literally ubiquitous vaginal bacterium, it wasn't even recognized as a lactobacillus and described as a species until 1999. And there were just very few isolates, and people who were studying it experimentally were usually growing it in a different media than they were growing all of their other lactobacilli, which, as you can imagine, is not really conducive to doing the best type of science when you're comparing different lactobacilli. And so I hypothesized that if we could understand why iners wasn't growing in conditions when other lactobacilli were, and if we could identify the unique metabolic and growth features that it possesses, we might be able to target that in ways that would differentiate it from other lactobacilli that seem to have better health associations. And so cutting out the vast majority of this paper, which feel free to go look up. I love it, but it's published, so you can go look at it. We figured out how to grow Lactobacillus iners in Lactobacillus MRS media. We figured out that we could add certain additives. We eventually narrowed this down primarily to the need to add cysteine. And here I'm just showing you an example. We have Lactobacillus crispatus grows well regardless of whether or not you put in additives. Lactobacillus iners not growing unless we add the additives. And then Gardnerella vaginalis, which doesn't grow in this media at all regardless of whether we put in the additives. So that was with an experimental strain, but obviously, you know, what you grow in the lab may or may not be representative of what's out there in the wild. So we took a lot of time to really generate a very large isolate and genome collection, including metagenome assembled genome. So this isn't solely biased by isolation bias for the different bacterial strains, representing diverse parts of the world. So we cultured bacteria from samples in South Africa, which was the first isolates from South Africa of these vaginal lactobacilli, but also the US. Genomes from Sweden, Italy, and China. And we kind of built this big species tree, and then we selected isolates from around the tree representing the diversity of the species, and we confirmed that we could see the same phenotype across all of them. We then looked at the genomes, and we found that we believed that this cysteine dependence could probably be explained by the fact that Lactobacillus iners lacks many cysteine uptake mechanisms that other vaginal lactobacilli possess, and therefore we tested this by treating it with cystine uptake inhibitors that had been described in other bacteria. And what we could see is that we could selectively inhibit Lactobacillus iners with cystine uptake inhibitors and yet not affect these other vaginal lactobacilli in culture. There's a lot of other work in here that I won't get into. So this was exciting, but if you look actually at the millimolar concentrations, this is not a very potent inhibitor. So there are some reasons to think this may not be the absolute solution for something you would run around treating people with. And we had other inhibitors that were more potent, but probably have toxicity issues. So this was very exciting. It was a great proof of principle. It wasn't necessarily the answer that we're looking for in the end. But it does highlight the possibility. We thought we could start going about and identifying using this type of method, ability to selectively inhibit L-iners. And so this brings me to what I'm going to spend the rest of the talk talking about, because while this paper was still a preprint, I got an email from Meilin, who said, I'm a graduate student, a PhD student at the Broad Institute, which is very close to the Ragon Institute. I'm an MIT biological engineering student, and I'd really like to spend the second half of my thesis working with you on looking at vaginal bacterial metabolic requirements. Because Meilin was really fascinated by the idea that you could use metabolites rather than simply antibiotics to either selectively promote growth of Lactobacillus crispatus or selectively inhibit the growth of Lactobacillus iners. So I didn't have an MIT affiliation, so we set up a structure where Meilin continued to be mentored by Paul Blaney, her PhD advisor at the Broad Institute, by my postdoctoral mentor, Doug Kwan, and then I served as her primary scientific mentor on the project that I'm about to tell you about. And then just to, again, highlight some other people involved. I think I've already introduced Jawu, Selena, and Nandu, but Jay, who's current technician in the lab from Mozambique, who grew up in Eswatini and is now a technician in the lab getting ready to apply to graduate school. So for reasons that are fascinating, but that I don't necessarily have time to get into unless we have time in the question and answer, Meilin really narrowed in on looking at oleic acid as a metabolite of interest. Also some related other long chain fatty acids. And so she tried treating cultures of vaginal lactobacilli with oleic acid to see what would happen. And what happened here is that it potently, far more potently than these other things I was telling you about, inhibited Lactobacillus iners and had no effect on these non-iners lactobacilli, which included many strains of Lactobacillus crispatus, Lactobacillus gasseri, Jensenii, and mulieris. This is in this media we had developed that was really optimized for Lactobacillus growth. But then we took this a step further because we'd also developed some media that could support the growth of these things, including Lactobacillus iners, but they didn't do that well in the base media, even though it's a very rich media, unless they had some additional metabolic help. And so she tried adding oleic acid to that. And remarkably, the same molecule robustly promoted the growth of these non-iners vaginal lactobacilli and continued to inhibit Lactobacillus iners. So the same molecule is inhibiting L-iners and enhancing the growth of Lactobacillus crispatus and related bacteria. And actually, this is a bactericidal effect on Lactobacillus iners. So here's some electron microscopy. So we have Lactobacillus crispatus across the top that was either untreated or treated with high levels of oleic acid, and then Lactobacillus iners that was either untreated or treated with high levels of oleic acid. And you can see that within 1 hour of treatment, there was just catastrophic cell and membrane disruption of Lactobacillus iners by oleic acid. And we've also done killing assays and ATP release assays to kind of confirm this phenomenon. So, okay, this was really shocking, exciting, if not unbelievable. We had a single molecule here that could selectively inhibit L-iners and selectively promote L. crispatus. And so that's great, but it's arguably not enough in and of itself, right? The then a miracle occurs phase of scientific discovery is where we were at. And so we had to ask the question, how and why is this happening? Can we really understand what's going on here? There's resistance to oleic acid and then growth promotion in these non-iners vaginal lactobacilli. And so the approach Meilin took to this is she said, let's take a look at the transcriptional response of these bacteria when they are exposed to high levels of oleic acid. So she grew these non-iners lactobacilli And she either did or did not treat them for an hour with high concentrations of oleic acid, and then she did transcriptomics. And so we have Lactobacillus crispatus, Gasseri, and Jensenii, and 3 genes that jumped out of us in the analysis of these were that there was an oleate hydratase upregulated in response to oleic acid. There was a putative fatty acid efflux pump, FAR-E, upregulated in response to oleic acid, and then there was a putative regulator of FAR-E that was also upregulated. And this is not, just to be clear, this was not us cherry picking these genes because they sounded like they were relevant, because actually, if you took the overlap of all of the differentially expressed genes across these 3 species, the only gene functions, I should say, across all these species that were differentially regulated was the upregulation of these 3 genes. So this is the overlap. This is the core set that they share. And when we treated them with linoleic acid or palmitoleic acid, which are other long-chain fatty acids, again, these 3 genes jumped out in terms of the overlap between these species. So this is very exciting. It's very suggestive. So we said, again, these are our experimental strains, which is great, but what about kind of in the broad field of these vaginal lactobacilli? Well, fortunately, we had generated these huge genome collections, and we could look to see for the presence of these genes. For example, we hadn't looked for the regulation in iners, because when you treat iners with oleic acid, it dies, which is not very conducive to transcriptional analysis, right? So we went to look for these genes, and what we found was really quite striking. So we have Lactobacillus crispatus, Gasseri, iners, Jensenii, and mulieris, and here we have OHYA and FAR-E, predicted gene functions. Iners is completely lacking it, and the others have it at the species level, and these are hundreds of genomes. And this is in contrast to other genes that are involved in the use of exogenous fatty acids, which iners does possess, as do these other bacteria. So the presence of these genes in all of these other bacteria is associated with the phenotypes that we had described. And in fact, this extends beyond something that you see just in the vaginal environment, because we pulled down the genomes. There was this recent systematic publication on the entire family Lactobacilliaceae, and Meilin pulled all the genomes, and we looked for the presence of OHYA and FAR-E in all of these Lactobacilliaceae genomes. And they were widely distributed. They were not in every species, but if we looked within the genus Lactobacillus, and sorry, this is an enormous figure, so it's not rendering very well. If we looked within the genus Lactobacillus, we had annotations of which of these species were associated with a vertebrate host. So not just humans. It could be humans, human vagina, human gut, human oral microbiome. It could be poultry, any vertebrate associated. Every single vertebrate associated Lactobacillus other than L. iners has both FAR-E and OHYA predicted in its genome. So there's something about these genes that seems like it's probably important, and L. iners is really the outlier here. And we did a lot of phylogeny on the genes, which is very fascinating. I'm not going to talk about it right now in the interest of time. But basically, getting more functional, we then took the genes and working with the Rock Lab at St. Jude Children's Hospital in Tennessee, we confirmed that this genomically predicted enzymatic activity was actually true. So the upregulated genes are indeed oleate hydratases. They take oleic acid is an 18-carbon long-chain fatty acid with a cis-9 double bond, and they hydrate the double bond to produce 10-hydroxystearic acid, or 10-HSA. And we confirmed that they actually do this biochemically. And then we said, okay, that's with the gene, the enzyme, right? Does this happen when you're actually growing the bacteria? So we did an isotopic tracing experiment with colleagues at the Broad Metabolomics platform, where we fed the bacteria labeled oleic acid and saw if it got converted into this molecule. And indeed it does. So here we're looking at Lactobacillus crispatus, Gasseri, and Jensenii cultures, looking in the supernatants. If we give them high levels of labeled OA, we get high levels of labeled 10-HSA out the other end. So when you grow them in culture, they're doing this. Now, we couldn't do this with L. iners at high levels of oleic acid because it dies. But if we grew it at a sublethal concentration in parallel with Lactobacillus crispatus, we could see even at low levels, Lactobacillus crispatus is still robustly producing this molecule, and L. iners is not, consistent with the absence of this gene on genome analysis. And then we wanted to ask, well, are these enzymes actually active where we really care about them in the vaginal microbiota? And so we said, well, humans do not have this enzyme. So we're not going to be making 10-hydroxystearic acid. And in almost all cases, or essentially all cases that we've looked, these BV-associated bacteria don't have oleate hydratases. So let's just measure this metabolite in human cervicovaginal secretions from a large set of samples, again, from the fresh cohort here. And what we saw is just really remarkable. 10-hydroxystearic acid essentially uniquely separates the samples that are in cervicotype 1 from all other samples, consistent with very high level activity of this enzyme within the vaginal microbiota. So this is really exciting. However, if you think very hard about everything that I just showed you, this is technically still all association. We haven't proven mechanism. We've just developed a very thoughtful hypothesis about the fact that the one or both of these genes is probably mediating mechanistically the phenomenon we've seen. And so this is where Meilin and I had a chat about this, and I said, you know, this is a really good project you've done already. You know, this is highly publishable, but if we really want to nail this, we need to do bacterial genetics. We need to knock these genes out of one of these vaginal bacteria for which we didn't have established genetics. And she decided to do it, which is amazing. And so the next thing I'm going to show you, it took about 8 months of work because she resuscitated an old genetic manipulation protocol that had been published for Lactobacillus gasseri for people who are working kind of in agriculture and the food industry, not focused on the vaginal microbiome. She had to regenerate all of the strains, all of the reagents, improve on the protocols, and so on. But what she was able to do is make clean genetic knockouts in Lactobacillus gasseri, which we had shown phylogenetically these genes look essentially identical and functionally identical as they do in Lactobacillus crispatus. And so just to walk this through for those of you who don't think about bacterial genetics very often, she made clean knockouts by double homologous recombination of the gene of interest. And then she was able to complement plasmid-based complementation and overexpression of the gene to confirm the phenotype if there was a phenotype with the knockout. And so moving ahead, we look to say, how does this affect oleic acid resistance in Lactobacillus gasseri? And what we saw is that FAR-E, this fatty acid efflux pump, was absolutely required for oleic acid resistance in Lactobacillus gasseri. If we knocked it out, they became susceptible to inhibition by oleic acid, and if we complemented it on a plasmid, we fully recovered the phenotype. And we also were able to show that the enzymatic activity when she knocked out OHYA went away. So OHYA, the gene, is mediating the product that we were seeing, as one would hypothesize. And when we knocked it out, there was no 10-hydroxystearic acid being produced. And when we complemented the gene, now there was being produced. If we knocked out FAR-E, it could kind of tweak the levels of 10-hydroxystearic acid production, but it didn't eliminate it. So that was FAR-E as a mechanism of resistance. What about a mechanism of growth promotion? Like, why would these other bacteria benefit from the oleic acid? And conceptually, you can envision that they were probably using it for something. That would seem to be a logical hypothesis. Sorry, I think someone is unmuted on the Zoom. Okay. And so we said the logical things that you would potentially do with a fatty acid would be that you would make membrane, or that you would use it for energy metabolism, you know, break it down by beta oxidation. So reviewing the genomes, it turns out that none of these bacterial species seem like they can make their own fatty acids. So we would predict that they would be entirely dependent on exogenous fatty acids for fatty acid metabolism. And they lack the genes for beta oxidation, so they should not be able to break these things down for energy. And so to test this, you know, we could grow them in lipid-depleted media and see if we got growth. And we didn't get growth in the FAR-E knockout, but it was dead, so that's kind of hard to distinguish growth promotion from resistance here. But if we looked at isotopic tracing, where we fed these bacteria oleic acid, we could show that they were using the oleic acid, the labeled oleic acid that we gave them went into building membrane. And it did not go into central energy metabolism. So they were using the oleic acid to build membrane. In fact, even L-iners does this, but only at very low concentrations, because otherwise it dies if you get to a high concentration. So these bacteria required the exogenous fatty acid. They could use it to build membrane. Now, if we think back to what I was telling you earlier, this presents a bit of a question, because there's nothing about oleate hydratase that's supposed to be involved in the process of building membrane phospholipids. And so we kind of had this model where you could take up the unsaturated long-chain fatty acid like oleic acid. You could use it to make membrane. You couldn't make your own fatty acids. And OHYA could convert it to these hydroxy fatty acids, but how does that help the bacteria? Because one of the key principles of bacterial evolution is if the gene is not doing anything useful, you're not going to maintain it. And yet that doesn't seem to be the case here. And yet we know that these bacteria are doing this, and they're doing it within the vaginal microbiota, because we saw this when we looked at the samples. And so we said to ourselves, we sat down and thought very hard about it, and we said, what could be the explanation for how this could offer a benefit? And I'm going to propose to you that we have found an answer, which is we thought, you know, these enzymes, this is not an energy consuming step. These are supposed to be bidirectional enzymes. And so we said, what if you have OHYA, once you land in a high OHYA environment, you have a pool of nutrients that no other bacteria except yourselves can leverage for membrane synthesis. And so we were able to show a couple of weeks ago by this complicated isotopic tracing experiment that indeed this enzyme can and does work in reverse. If we culture the bacteria with OHYA and OA together, they can still use the 10-hydroxystearic acid, sorry, that's what I should have said, for membrane synthesis. And then if we grow Lactobacillus iners or these other lactobacilli in 10-hydroxystearic acid instead of oleic acid, indeed, in lipid-depleted media, indeed 10-hydroxystearic acid is capable of promoting the growth of these lactobacilli. And if we do it in the knockouts, we can actually see that this is entirely dependent on the presence of this enzyme. And in fact, if we do competitions where there's no antibiotic to preserve the gene or the enzyme, the gene is entirely selected for in a 10-HSA enriched environment. So to summarize what we have here, we have an intervention that seems to selectively inhibit Lactobacillus iners, to selectively promote Lactobacillus crispatus, and we think we know why. And so this is really interesting for a candidate for treatment. Remarkably, and I'm going to go through this quickly because we're running out of time, this molecule also inhibits many BV-associated bacteria, including some that are metronidazole resistant, which I kind of couldn't believe when we got the result, except it's real. And so now we've actually checked off all 3 of the items on our wanted poster. And so what we did is we developed this system. We don't have an animal model, but we generated in vitro defined BV-like bacterial communities that have a mixture of bacteria and a mixture like what you might see in someone with BV. So these are single strains mixed together. We then grow them and control metronidazole, oleic acid, or oleic acid plus metronidazole, and we see what happens. And after culturing these are multiple replicates we're looking here. So if you culture these without oleic acid or metronidazole, you basically preserve this BV-like community composition. If we add metronidazole, we see a predominance of Lactobacillus iners, as one would typically see in someone treated for bacterial vaginosis. If we add oleic acid alone, we get an enrichment of Lactobacillus crispatus. We get rid of our iners, but there are some BV-associated bacteria, some Prevotella, that do fine with oleic acid. And if we add metronidazole, we get near complete dominance of Lactobacillus crispatus. And we can quantify this by the ratio of Lactobacillus crispatus to everything else, and it's highly significant. And when Mei-Lin did the same experiments where she added 10-hydroxystearic acid instead of OA, sorry, this didn't render very well, she gets a similar phenomenon. So, in summary, I'm just going to skip ahead here, sorry, since we're running out of time. What we can say overall is that the vaginal microbiota is linked to BV, to cervicovaginal health and disease risk. L. iners dominance has less optimal health associations than L. crispatus, but VV treatment tends to promote L. iners instead of crispatus. If you give people a live biotherapeutic, it can improve BV treatment, but there are issues with partial efficacy because Lactobacillus crispatus seems to not often be colonizing. We can leverage cysteine and fatty acid metabolism to selectively inhibit L. iners growth and or to promote Lactobacillus crispatus, at least in vitro. And these findings suggest novel approaches for improving BV therapy. And finally, at a broader level, this suggests that continuing to study bacterial growth and metabolism can allow us to identify determinants of microbiota composition and potentially additional new potential treatment modalities. So with that, I'm going to thank many, many people who've helped me with this, mentored me, been wonderful collaborators, particularly my postdoctoral mentor, Doug Kwan, and many, many wonderful people who worked on this project. And thank you. And sorry, I went right to the end of time.
Thank you so much, Seth. I'm being a terrible host. I shouldn't have given such a long introduction, but we do have a couple of minutes. If you can stay along online for some questions for Seth, please do. I'm going to open the floor now for questions. Today looks like it's the first one.
No. And just to add, yeah, we have a lot of high-level seminars, and one thing that we normally do is to grill back the presenters. So do not be shy. Yeah, people that have to go can go, but please, yeah, let's engage and let's ask difficult questions. Yeah. Good.
We need revenge for me taking Lenine up Table Mountain here. So.
Yeah, because thank you. So we have here where's the 2, 3, 4. Okay.
Thank you. It was a fascinating talk. Really good work. I just want to go back to your introduction. That this first slide where the Peebles diagram showed some figures that I couldn't really understand. I assumed that was incidence, but it doesn't link to what you described there. So I'd be interested to know.
Sorry, this is about with respect.
It's the introduction. Where you talked about incidence of BV.
Let's see. This one or?
No, totally at the beginning. In your introduction. Yes.
This one?
Back. There you go. Peebles.
Yes.
What are those percentages there?
So the percentages here, yes, this is, I should say, this was an analysis kind of everything in the literature. There have been other studies that came to slightly different conclusions about this, but basically the numbers here are estimates from many different studies around the world about prevalence of bacterial vaginosis in different populations. And I think what you may be highlighting is that for some of these, I made a comment that this seems to disproportionately affect in a number of studies members of many African populations. The Peebles analysis, I will say, saw this a little less so. However, there have been other analyses that have suggested this to be the case. Whether this is because the study selection for many of the studies that have been done in Southern and Eastern Africa have particularly focused on people who are otherwise known to be high risk for things like HIV, and therefore there's kind of a selection bias, or whether people's found some studies that maybe were biased in another way is a little unclear. I can certainly say that for many of the other studies we've done, and I think, for example, Joanne and Lenine have seen this in their studies as well, the rate of BV is often higher than 25%, certainly in a South African setting in many of the studies that we're most closely familiar with, I would say. I don't know if Joanne or Lenine have additional comments on that.
Yeah, thanks for that. It was quite unclear because of the disproportionate effect. It doesn't look like that on the figure, so
yes.
It would be good to explain that. Thank you.
Hi, so I just wanted to find out about washing or hygiene practices, if those do influence the vaginal microbiome, and then if it does, are the studies that are looking at treatments keeping that as a factor? Are they implementing specific soaps or things for people to use, and do people continue to use those afterwards, or are those kind of things that could wipe out your beautiful treatment that you're proposing?
So the question for anyone who didn't hear it is about whether intravaginal hygiene practices of various sorts is associated with or causative of changes in microbiota composition. There is a clear association in a variety of studies of douching, or particularly in a variety of cultures. Within Africa, there's been a lot of research looking at the vaginal drying agents that are used in some communities. There's a clear association of that with bacterial vaginosis. And it's extremely plausible and, if not likely, that there can be a causative effect there, although you may be more likely to use a drying agent if you're having vaginal discharge, so there may be a little bit of a circular argument there. I mean, in general, I think what most experts in the field would say that the best intravaginal hygiene practices is one that tries to leave the vagina alone rather than putting lots of soaps and lotions and who knows what in there. But different people do different things. There are different cultures and different personal practices. And those may or probably do influence. It's hard to imagine that they wouldn't influence the microbiota. And so, yes, and sex itself. I mean, there's many other things. Yes, and Joanne has a comment. I'm reciting her research, so she could probably explain this better than me.
Seth, this is brilliant, and I think the defining thing with Malin's study, so congratulations, was the Cox postulates and like taking it beyond association and doing the knockdown and knock-in and everything. And I really think that differentiates it. So I look forward to seeing the paper being published. And I wasn't going to comment on vaginal practices at all. I'm actually just curious about like what is the road to market and where do you take this information now, and what's the next trial look like for you guys to investigate this? And then the second thing was somebody at a meeting we were both at last year brought up the fact that a lot of these fatty acids or oils are not great for condoms. So whether that had factored into where you were thinking about going into treatments adjustment.
Yeah, so these are all, thank you, Joanne. These are all great questions. So what would be a theoretical path to actually translating these basic findings into treatment? It seems to us, I assume it would seem to many of the rest of you, that this is something where a topical application could make sense or would be the most logical route. Oleic acid, since it's in things we eat and so on, is in a category considered generally regarded as safe, but I think nonetheless it would be important to do some basic toxicity testing above and beyond anything we've done, make sure it doesn't cause a massive overgrowth of like vaginal candida, things like this that we haven't yet assessed. There is also an issue that if people were to think about administering this to themselves, I mean, we're not supposed to use oil-based lubricants or probably fatty acids with condoms, right? Because it can destroy the condom integrity. And so that would definitely be a consideration. But the theoretical path to what this would look like, I think, would be probably, I think, a combination of an antibiotic such as metronidazole, a Lactobacillus crispatus live biotherapeutic, and then adjunctively, concurrently with that or soon thereafter, some oleic acid. Now, whether that would need to be long-term or whether that's just what you need to give Lactobacillus crispatus its foothold, at some point, the bacteria, the Lactobacillus crispatus, is going to have to succeed on its own. Like, you can't just help it every day, probably. But that would be, I think, where this would be going in terms of treatment and prevention of recurrence. That would be the hope, but we haven't done any of that yet.
Hi. Just a quick question on the oleic acid, because I was too amazed to take it in. Is this just that the bacteria are utilizing the oleic acid? There's no production?
Yes, that's correct. So these bacteria are fatty acid oxytrophs. So they can't make their own fatty acids. They lack the genes, and if we grow them with precursors like acetate, they can't grow in lipid-depleted media. So they are not making their own oleic acid. And we've also just confirmed this by culturing them and seeing that they fail to produce it.
Okay. I also think it's very interesting whenever I look at research that's on live biotherapeutics, I don't think I've ever come across anywhere where people have measured adherence to live biotherapeutic products, because the endpoint that you're measuring is actually also kind of potentially what would be showing that you have adherence. So how does one separate colonization as a result purely of adherence to the treatment versus some other mechanism?
Yeah, that's actually among the data I didn't show you. So for the Lactin-V study looking at adherence, participants in this study, kept very detailed logs, which we all know that self-reported adherence has its pluses and minuses, but they also returned the applicators to the study clinics, and those could be stained for markers of vaginal fluid, so to actually confirm that there was evidence of adherence. And both in the New England Journal study for recurrent BV and also what we see for Lactobacillus crispatus colonization, there is a clear signal that higher adherence correlated with better outcomes, be that in terms of clinical BV or be that in terms of Lactobacillus crispatus colonization. But it's also clear that people who definitely seem to have been using the product did not necessarily, in many cases, get colonized or prevent recurrent BV. Great question.
Hi.
Hi.
No, go ahead.
Hi there. Thanks for the talk. Just one last quick question. I was wondering if you had any intention of manipulating L-iners specifically, genetically manipulating that and doing knockouts on iners.
Theoretical intentions or actual intentions. So one of the few labs that had really done very detailed experimental work on Lactobacillus iners prior to this was Adam Ratner's lab. This was the lab that discovered this cytolysin that is a putative virulence factor within Lactobacillus iners. And if you look at his PhD student's thesis, he has an entire appendix about how he spent what sounds like about 1.5 years of his life trying to genetically manipulate Lactobacillus iners without any success in transforming it. So I think this would be great. I would love to look at it at some point. I am not actively doing the experiments at the moment because I need kind of the right setting and the right person to work on it.
Okay, thank you.
So we have a question over here.
Yeah. Are you offering to be the right person to
Yeah.
work on that?
Give me a call. Yeah.
Do you want to waste a year of your life? Questions, right? And Tulio, and 2 questions online.
Hi, Seth. Excellent talk. I'm thinking about stepping away from interventions. I'm thinking that you have access to all these genomes and knockdown phenotypes. Is it now useful going back to the initial 16S work that has been done to add in additional targets and maybe long-read sequencing to improve the characterization and detection? Because I know there's high similarity of 16S between different bacterial species. So do you think there's a chance of false positive results?
False positive with respect to what? Just, it's a good question, but just make sure I'm answering it.
So in terms of it coming up on a database as the target, but it might not honestly be that. And even though an intervention downstream would be identified, it might be to something relatively closely related to what you initially targeted. You
mean that what we see by 16S might not actually be the genus or the taxon or whatever?
So adding targets from what you know now,
yeah,
and also looking at the entire genes and not snippets.
Yeah. So this is a good question. I mean, one of the major challenges in this field from a bioinformatic perspective is that a lot of the bacterial metagenomics tools and databases and so on that have been developed were not informed by vaginal microbiota research. And so constantly in this field, we see papers by people who did their 16S sequencing according to standard procedures, did their annotations using a completely standard database that you would use for all your gut microbiome work, and get the wrong answer because the databases get it wrong, even at the 16S level. And so what we've really found is that there's a lot of things that are either misannotated or not annotated well, even at the 16S level. When you get to the shotgun metagenomics level, this is in the process, I think, of starting to improve, and we've tried to do some work to leverage this, although it's not still fully together. The genes that are present in these pangenomes are not really well characterized, right? And so by generating some of these species level genomes, pangenomes, I think it'll give the opportunity, although this hasn't fully iteratively happened, to better understand the taxonomy. Because one of the issues with bacterial shotgun metagenomics, obviously, is that if you see a gene you know comes from a certain species, that's fine. But if you see a gene that you don't recognize in your database, it becomes very confusing how to interpret it. There is also a tool that looks at a gene-based analysis rather than a species-based analysis developed by Jacques Ravel's lab called Virgo, which is also very exciting. I don't know if I'm answering your question, but I mean, these are some of the challenges that currently exist in the field that we and others have been trying to address.
So that's the same with TB, for example.
Same with
for tuberculosis research, for example.
Right.
A lot of people will use 16S, but it's so conserved amongst different, over 180 different members of the genus that you might not be detecting what you think you're detecting.
Yeah, I mean, one advantage we have is, I mean, there's certain taxa, I mean, these aren't, all within the same genus, right, for example. So for the lactobacilli, there can be issues. Like we can't tell from standard 16S sequencing which one is Lactobacillus miliaris versus Jensenii. They're closely related species, but they're clearly distinct. And that's a big problem with some of the older data. And you really need shotgun metagenomics or other techniques to do that. But telling that someone has a lot of Prevotella versus a lot of Lactobacillus iners, like I think we're getting that right for sure,
Yeah, thank you. And sorry for everyone giving you a hard time. Yeah, but just to say that it's absolute phenomenal, not only the results, but how you guys start for doing genomics, and then after you did cell culture, and then you did for RNA-seq expression to identify the genes, and then went to the proteomics, and then in the end did like some, knock out some genes and grow the thing. So my question to you, like, how did you manage to kind of collaborate with all these different disciplines? Yeah. And how did you manage to do that, not only between only the MGH, but also sometime approaching the MIT and Harvard? Yeah. And how do you guys suggest this collaborative program that we so much need to develop here in South Africa? So, for example, it's great to see some people from UCT and Stellenbosch, but that's something that we need to do, is to learn to work across disciplines to answer a question. Yeah.
So the question is about how does one think about approaching the collaborations and using so many modalities? Obviously, I'm showing you the examples where that's been successful, not the ones where it hasn't, of course. So there's a survivorship bias in terms of what I present. But I think the big thing is if you're running the project, like what we've really tried to do here, because like I said, for example, we don't have an animal model, right? And so there are limitations. I mean, there's obvious experiments we should be doing, except nobody, not just us, has the ability to do those experiments right now. Or, we should be cloning these things into L-iners, except right now those technology don't exist. So is really thinking to oneself, what is it that I would be doing in this situation if I could, acknowledging that I am not an expert in targeted lipidomics, for example? Right? What is it that in an ideal world I would do here if I had an animal model? And I can't, but at least I can ask questions about what I should see if I were to do it in an animal model, things like that. So in this particular example, we were fortunate. I mean, first off, I have spent a lot of time trying to teach myself both the bacteriology, which is what my PhD was in, and then various of the bioinformatic techniques, and really just spent a lot of time delving into those, doing the 16S analysis, epidemiology training as a physician, and so on. But the second thing is that we were able to structure our questions carefully enough that in some cases we were just able to identify papers that it turned out, may have been published 20 years ago, but did what we want to do and dig up the protocols because we really refined for ourselves what we were looking for. And then the other thing was, like, for example, going to the Rock lab at St. Jude Children's Hospital, we had figured out it looks like we should be studying Oleic hydratases. And we were able to schedule a meeting with Dr. Rock, who unfortunately very tragically passed away late last year. But we were able to structure a meeting with him, one of his senior postdocs who's now at University of Kentucky, and their senior staff scientist, and really come in with a very focused set of questions. And the official ask was, could you lend us some of your reagents and so on? But they were so fascinated they'd been studying this enzyme in Staph aureus, and they were so fascinated by having it in a different field that it led to them being interested in a collaboration. Similarly, with the Broad metabolomics platform, they were so interested in the project because we gave them a very clear pitch that they actually didn't even charge us anything. They just did it as a collaboration, and they're on the paper as authors now. And so part of it is we're very fortunate, in Boston to be in an environment where there are a lot of resources around, and that obviously helps. But the people at St. Jude's are very, very far away, and they got interested in the science because we were able to articulate what is it about this research that fits with your own and come on board our team.
Could I add to that? I've actually seen this in action. So having your work presented so often to a diverse group of people, as we have over here, I've seen that with your work. Make new collaborations, make new networks of people who are interested in addressing similar problems from varied angles. So well done for you and your team and your networks and all this amazing work that you've been showing us in the future for genital health. Well done, Seth. Thank you very much.
Thank you, everyone. Great questions.